So let's continue our discussion on microbial genetics with a discussion on how we can mix and match DNA molecules, or mix and match the genomes of our microorganisms. When we have recombination, we are going to have effectively the bacterial version of sex. Now, obviously, we don't have like males and female bacteria, but we can exchange bacterial information, or excuse me, genetic information between bacteria. This is known as recombination. Generally speaking, during recombination, bacteria A donates some DNA to bacteria B, to another bacterium, and the end result is this bacterium that received the donor DNA is going to be changed. It'll be different. We've made it better. This is a key way that we can transfer antimicrobial resistance from one bacterium to another bacterium. Usually when this transformation is going to occur, or recombination I should say is occurring, it's going to occur when there's a sex pilus, which is a small tube, attaching the two bacterium, and then a plasmid, that small circular piece of DNA, is moved through that sex pilus from the cytoplasm of bacterium A to the cytoplasm of bacterium B. This gives up genes, as I just mentioned, resistance to drugs or metabolic poisons, or some other new metabolic capability. When we look at an organism that contains genetic information from another organism, a generic term for that is recombinant. So if recombination occurs, the end byproduct, or that end organism, is called a recombinant. So first let's focus on our plasmids. This is a small circular piece of DNA. Each plasmid contains its own ori, or own origin of replication, usually referred to as the ORI, the ori region. They replicate independently of that main bacterial chromosome. The plasmids are not necessary for survival. The bacterial chromosomes, the big chunk of DNA that contains all of the vital genes to keep the organism alive. The plasmid contains the accessory genes that give those exciting new traits to the organism, like ampicillin resistance, or whether or not to produce cilia, so on and so forth. Sometimes they carry useful traits, sometimes they just carry stupid traits, and they don't really confer a lot of benefit to the microorganism. Other times, if we aren't transferring a plasmid from organism A to organism B, we could transfer a chunk of the chromosome. Sometimes we can just slice out a piece of the bacterial chromosome and then send it to another cell and insert it in that second bacterial cell's chromosome. That's a little bit more complicated. We'll talk about how that happens. So, let's talk about the three primary modes of gene transfer. Remember, if we mix and match DNA from organism A to organism B, it's recombination. Recombination can result in, or a kind of recombination is horizontal gene transfer. Horizontal gene transfer, by definition, is within a generation. It's not from a parent to offspring, but it's from a sibling to sibling, so to speak, within a generation. So there was no reproduction that occurred. We can have horizontal gene transfer occurring with conjugation, transformation, or transduction. During conjugation, the two cells are physically connected with a tube. This is directly from cell A to cell B. This is what I've talked about primarily. We also have transformation. During transformation, cell A will squirt out some pieces of DNA. That piece of DNA floats around in the extracellular environment, and then cell B sucks up that piece of DNA that's floating around in the extracellular environment. And then during transduction, a piece of DNA from the donor cell is removed by a virus that affects bacteria, aka a bacteriophage, and then that bacteriophage serves as a vector, or an intermediate carrier, that will carry the genetic information from cell A to cell B. So, in example one with conjugation, we have direct linkage between the cell. We need to have that sex pili form, so to speak. During mode number 2, during transformation, the cell A, the first cell, the donor cell, will exocytize, or it will squirt out its piece of DNA, and then cell B will randomly find the piece of DNA and suck it up and incorporate it. And then during the third method, during transduction, we have a virus transmitting the information from the donor to the recipient cell. So let's talk about conjugation, where they bind together. This one can be relatively complicated depending on how we cause this binding to happen and what kinds of cells are binding. Let's think back to gram-negative and gram-positive cells. It's been a while now. That was several chapters ago. Gram-negative cells have a cell membrane, a cell wall, and then another cell membrane. So there's a cell membrane sandwich with that peptidoglycan cell wall in the middle. So gram-negative bacteria, generally speaking, are going to have different mechanisms in play compared to the gram-positive bacterium that have just a cell membrane and the cell wall. So let's look at how we can have conjugation in a gram-negative bacterium. Then we'll focus on our gram-positive bacterium. For gram-negative conjugation, we need to have that the F prime factor, or the fertility factor. This F prime factor will allow for the bacterium to make the sex pilus, or the conjugated pilus. And this recipient cell, the recipient cell, that is also going to be gram-negative, will need to have a recognition site. So we need to have the donor appendage and the receiving, or the receptor appendage, so to speak, for these two gram-negative bacteria to link up. For the for the period that is going to make that, sorry, I just totally lost my train of thought right there. Someone just went by in the background. You can't see that on this recording, though. I digress. So, for the cell that has that fertility factor, it's F positive. It has the plasmid that allows for the gram-negative bacterium to make the conjugative, or sex pilus. If the cell is F minus, it doesn't have that plasmid, so it does not have the ability to make the conjugated pilus at that time until it receives the plasmid, and then it can start making the conjugated pilus. So, let's say we have a F-positive cell. That F-positive cell grows essentially the sex pilus, or the conjugated pilus, and that pilus docks or attaches to the F-negative cell. That F-negative cell is then going to be drawn towards the F-positive cell. The two cells are then drawn together, and genetic information can move from cell A to cell B, from the donor to the recipient. So here's the F-positive cell, here's the F-negative cell. The F-positive cell, boom, attaches a sex pilus, boom, right there. And then the plasmid, the F-vector plasmid, can reproduce, and separately from the nuclear, or the bacterial chromosome, and transfer itself, the copy, into the recipient cell. And this recipient cell went from F minus to F positive. And while this pilus is attached, if there are any other plasmids present in the cell, maybe a plasmid that would code for, antibiotic resistance, that plasmid could also move through the sex pilus at the same time. This is a primary mechanism we're seeing right now in developing, or in third world countries, where we're seeing strains of antibiotic resistance that are coated on plasmids, moving from gram-negative cell to gram-negative cell, jumping across bacterial cell species, and then spreading rapidly throughout the native populations. This is the kind of stuff that keeps me up at night. I think, personally, as an aside, within 20 years, we are going to be in a post-antibiotic era, which is rather terrifying. So let's talk about how gram-positive cells combine together. We still are going to have that opening between the two adjacent cells. We're still going to have replicated DNA moving from one cell to another cell. The gram-positive conjugation is going to have a slightly different mechanism in play because of its slightly different composition of the cell envelope compared to the gram-negative bacterium. Conjugation between bacterium is always going to be a conservative process. The donor bacterium is always going to retain or keep an original copy of the genetic material that's being transferred. I shouldn't say necessarily an original copy, but it'll keep a complete copy of the genetic material that's being transferred. So here we have the donor cell, here we have the recipient cell. The donor cell here at the end always is going to keep a copy of the genetic information that it donates. In E. coli, that F factor will make a copy of itself, and that recipient cell receives the F factor, and then the recipient cell becomes F plus. It's now capable of making that sex pilus. Sometimes we'll see that we have HFR, or high-frequency recombination donors. These occur when the plasmid becomes integrated into the F-positive donor chromosome. So we'll have that plasmid for the F-positive factor, and it's going to be integrated into that donor's chromosome, so that it's now every daughter cell from that donor cell is going to be able to make its sex pilus. In other words, instead of having the F factor stuck in a plasmid that may or may not be in the daughter cell, that F factor gets moved into the bacterial chromosome. And while that F factor is in the bacterial chromosome, every single daughter cell that's produced through binary fission is going to have the F factor, and every daughter cell will be capable of making a sex pilus. So all of a sudden, that phenotypic trait becomes much more permanent in the population of bacteria. Oh, here's another figure to explain just what I talked about. Here we can have the F factor that's been integrated into the host chromosome. That F factor can be transferred with a sex pilus, and then the recipient cell has the F factor incorporated into its chromosome, and all daughter cells will now have the F factor. Sometimes the plasmids contain genes that confer resistance. Antibiotic resistance is one that is very well studied. There's a lot of incentive to study it right now, so we're going to focus the most on antibiotic resistance throughout the scope of this course. But there are other kinds of traits that can be conferred. There could be heavy metal resistance, maybe resistance to mercury, or copper, or silver, or they can confer a virulence factor. So now that bacterium can start to make adhesion molecules that help it be resistant to phagocytosis, or it can help that bacterium to make an extracellular enzyme or an exotoxin that will break down the extracellular environment and make it more favorable for the bacterium. Let's talk about transformation. So sucking up naked DNA from solution. Remember, there's 3 primary modes of horizontal transfer: conjugation, then we have transformation, then we have transduction. We've just talked about conjugation in detail with our F factors and our HFR factors. Now let's focus on transformation. So during transformation, that bacterium is going to suck up a piece of DNA from solution. Usually that piece of DNA solution comes from a different bacterium, a separate bacterium that has gone through apoptosis or cell lysis and has shot bits and pieces of its DNA into the solution. During this transformation process, it's going to be very nonspecific. The recipient cell is going to pick up all the little tiny bits and pieces of DNA that it can from its extracellular environment. Generally speaking, there are DNA binding proteins on the surface of the recipient cell that facilitates this transformation process. In order for a cell to receive DNA from the extracellular environment, that cell needs to be receptive. So only receptive cells can receive DNA. We call those receptive cells competent cells. Now, in nature, generally speaking, competent cells are going to be controlled by genes that will code for the DNA binding proteins on the cell wall or the cell surface. But in a laboratory environment, we can force non-competent, non-compliant cells to become competent cells. And this is a standard technique that I would do all the time. In the lab I worked in, we would use electric shock to take those negatively charged phosphate groups in the DNA backbones and shoot the phosphate backbones and the negatively charged DNA molecules into the bacterial cell wall and have the pieces of DNA perforate the bacterium and go into the cytoplasm. That was one way we could force the DNA into the bacterium. Another way we could do that is by taking the bacterium and putting it in a salty, soapy solution. Salty, soapy solutions make it easy for stuff to penetrate the cell envelope of bacteriums and other cells, and we'd take our DNA and mix it with the bacteria in the salty, soapy solution, and a lot of the bacteria would suck up the DNA fragments, or become competent. Transformation, the sucking up of extracellular DNA fragments, doesn't require any special appendages. It doesn't require the donor or recipient cells to be in direct contact. The donor cell ejects the DNA, and then random recipient cells will float up to the DNA. So, as we're going through biotech, and we'll talk more about biotech later on in this chapter, this is a very useful way to insert your favorite gene, whatever your favorite gene may be, into a microorganism, and then you can carry out experiments to see how that gene works. What does that gene make? Or you can isolate the product produced by that gene and purify it out for use in a laboratory setting. Transfection is another process that's very similar to transformation. Transfection, though, is the transformation equivalent for a eukaryotic cell. So transfection occurs when we will do this process to yeast or mice or plant cells. Sometimes for transfection we'll use a bacterium intermediate. Agrobacterium was a common intermediate that we used in the lab to insert our favorite gene into a eukaryotic organism. Let's talk about transduction. That's the third method. Recall, we had the three primary methods: conjugation, transformation, and transduction. Transduction is very specialized. It's going to involve the use of a virus intermediate or a virus carrier that will carry a chunk of the host genome to the recipient. And it will do that by taking that donor's genome and incorporating a chunk of the donor genome into the genome of the bacteriophage. And then when the bacteriophage infects a second cell, that second cell will receive a piece of the donor's genome when the bacteriophage inserts its genetic information into the recipient cell. So first, let's talk about our host cell. So the first starting cell has the bacteriophage infected. That bacteriophage then incorporates its genome into the main bacterial chromosome, and during this process of incorporating the bacteriophage genome into the host cell's genome, a bit of the host cell DNA gets mixed with the bacteriophage DNA. Later on, that prophage can be activated and carry a chunk of the host DNA with it. And when that bacteriophage enters the lytic cycle, that host DNA is going to be incorporated into the viral DNA and then carried to that second bacterial cell. So here is our host cell. It has some dark purple bacteriophage DNA mixed in with it. That bacteriophage DNA gets excised from the host nucleoid region containing its chromosome, and it carries a little bit of the host bacterium DNA with it. Then we have modified bacteriophages that have hybrid DNA of bacterial and viral DNA mixed together. That bacteriophage leaves the donor cell, binds to the recipient cell, and injects its genetic information to the donor cell, and then the donor cell receives bacteriophage DNA and bacterial DNA from the original host cell. Finally, let's talk about our jumping genes, our transposons. A transposon is a gene that can be cut and paste completely from one chromosome to another chromosome, from one part of the genome to another part of the genome. These jumping elements or jumping genes can move from one part of the genome to another part of the genome. They can go from a chromosome to a plasmid, back to the chromosome, or vice versa. They can go between bacteria and eukaryotic organisms. These are going to be the mixing and matching, or the accessorizing of genes within a fragment of DNA. Some transposons replicate themselves before jumping. For those of you that are interested in irreducibly complex molecular machines, most current theories that explain the origin of these irreducibly complex molecular machines involve transposons replicating themselves so that we have two copies of a gene, one that can be slowly modified over time, and then the original that can continue to carry out the vital life functions of the cell. And I'm starting on a tangent again. I'm stopping it. I'm nipping it in the bud. Back on task. Back on topic. So transposons can change the morphology of a bacterial species or a bacterial colony, pigmentation or antigenic characteristics. In other words, as we have genes jumping from bacterial species to bacterial species, we can change how those species act. That's no big surprise. If we change the programming of the cell, we can change the behavior of the cell. Transposons also function well for replacing damaged DNA within a section of the chromosome and can allow for drug resistance, of course, to transfer from one bacterium to another bacterium, propagating antimicrobial resistance throughout a species. So, here is our starting cell. We have a transposon, or jumping gene present in the circular bacterial chromosome, and that transposon can go from the top to the bottom. It can just jump from one part to the other part, or the transposon can copy itself and then appear in multiple spots in the bacterial chromosome, or the transposon can go from the chromosome to a plasmid. So the transposons can go all over the stinking place. There's no stopping them. So, concept check. Which of the following mechanisms involves horizontal transfer from bacterial DNA through a bacteriophage? Is it transposition, conjugation, transformation, transduction, or E, none of the above is correct? If you don't remember, you can flip through your notes, you can check your PowerPoint slides, you can rewind the video. Go ahead and pause it and find me the answer right now. 1, 2, 3, 4, 5. The correct answer is transduction. So D. If you have any questions about this recording or this section of your textbook, please post them on the discussion board or shoot me an email. Happy studies!